Hi, I'm Ben Yerys. Nancy, Lee, and I want to welcome you to our symposia on executive function and brain in everyday life. IDDs are associated with executive function weaknesses. And today, our research is going to, or our symposia is going to address two gaps in the research. One is going to be neural and real world correlates of executive function across multiple IDD groups. And the second is going to be how EF skills may play a role in how we are able to collect data from various IDD groups. As you can see here, we have 4 talks lined up for you today. Each one is going to last about 10 minutes. And then at the end, our discussant, Lauren Kenworthy, will sort of describe sort of key themes and future directions of research. And then we'll open up for questions at the very end. Thank you for coming, and we hope you enjoyed the symposium.
Be talking about the IQ adaptive functioning gap in autism spectrum disorder. In brief, here's an outline of what I'll be covering. I'll give some background to set up 2 studies and present data for one study that looks at the IQ adaptive functioning gap in autistic and typically developing youth, and then another examining the correlation between this gap with brain structure. As we all know, 2 critical components in the diagnosis of intellectual disability are intellectual functioning and adaptive skills. According to the CDC, approximately 2/3 of autistic individuals have an IQ greater than 70. In spite of these relatively high IQs, however, many do not ultimately attain the level of independent functioning we may expect given their intellectual ability. Therefore, the current talk will present results from 2 studies looking at the IQ adaptive functioning gap in autistic youth without co-occurring intellectual disability. Among autistic individuals without co-occurring ID, there is a gap between IQ and adaptive skills, and specifically IQ is much higher than adaptive skills. We don't know, however, whether this gap is associated with other key metrics of cognitive functioning, nor do we know what, if any, brain metrics this gap might be associated with in autism. Finally, no study has examined whether a similar gap between IQ and adaptive functioning is found in typically developing individuals. We will examine the gap as it relates to a key cognitive metric, executive functioning, or EF. EF is an umbrella term, and it's used to describe higher level cognitive skills that help us attain goals and solve problems. There's a rich literature documenting EF difficulties, especially in real world contexts across the lifespan in autism. The brief is a questionnaire that yields a real world assessment of EF. It's used both here in the current talk and in 2 of the upcoming talks. In the brief, an informant responds to questions according to a 3-point scale. The responses are never, sometimes, or often. Higher brief scores reflect more EF difficulties. The brief has 1 global score, and that global score is broken down into these 2 indices. Those indices, in turn, are broken down into subdomains. For example, the BRI is broken down into inhibition, shift, and emotional control. And these probe the overall ability of the individual to regulate and adjust behavioral and emotional responses in a way that is suitable for a given context. Likewise, the metacognition index is broken down into 5 subdomains, and these probe the ability to self-manage tasks and to engage in real-time performance monitoring. For the current study, we look at the gap between IQ and adaptive functioning in autism and also in typically developing youth, and examine whether this gap relates to EF, specifically the metacognition and behavioral regulation indices of the brief. The data we report on are from an ACE, or Autism Centers of Excellence network. Because the goal of this network grant is to examine sex differences and the female phenotype in autism, autistic females were oversampled. And so you'll notice a unique feature of the sample is that it is sex balanced. Our measure of adaptive functioning is the 2nd edition of the Vineland. Our measure of IQ is a differential ability scales using the general conceptual ability score. And of course, our measure of EF is the brief. To provide some context and to show that our sample is consistent with decades of research, we see that autistic youth show substantial adaptive skills challenges compared to typically developing youth. And this is true across the three subdomains of the violin: the communication, living skills, and socialization subdomains. Our first aim was to look at the gap between IQ and adaptive functioning, and a paired samples t-test revealed that both autistic and typically developing youth demonstrate this gap, showing significantly higher IQ relative to adaptive functioning across the three domains of the violin: communication, daily living skills, and socialization. Moreover, we find that this gap is positively associated with age in this cross-sectional sample, where older individuals have a larger gap between IQ and adaptive functioning. This age effect, consistent with our previous slide, is consistent with it being driven by quote-unquote decreases in adaptive functioning with age in the face of stable IQ scores. Importantly, returning to these results, though the typically developing youth do evidence a gap between IQ and adaptive functioning, and Mankova controlling for age revealed that this gap is significantly greater in the autistic sample. And so, as predicted and consistent with the literature, we do find this profound gap between IQ and adaptive skills in autism. And it's critical to note that these autistic youth have a mean IQ of 101, whereas the mean violin score is 73. So we're seeing an adaptive functioning score that averages 27.5 standard points lower than their IQ. In terms of understanding how this profound gap between IQ and adaptive functioning might be associated with other indices of cognitive functioning, partial correlations controlling for age show that in autistic youth, the gap is associated with EF problems as measured by the metacognition index of the brief. So a larger IQ adaptive functioning gap is associated with greater real-world problems in the domains of working memory, initiation, planning and organization, and monitoring. Next, we turn to an examination of the association between IQ adaptive functioning gap and brain structure, looking at two metrics of brain structure, namely cortical thickness and the structural covariance of cortical thickness, both of which I will define momentarily. We used a subsample of the participants from study 1 who had usable structural MRI data. And the samples in study 2 are quite similar to those from study 1 in terms of age, sex ratio, and IQ. We focus on two key brain-based metrics. First, cortical thickness itself, looking at the raw scan on the left and the blown-up version of this on the right. You can see the peel surface is in red and the gray matter, white matter boundary is in yellow. And so cortical thickness is simply the distance between the peel surface and the gray matter, white matter boundary. The algorithm used in the FreeSurfer software essentially creates a net of triangles across the entire brain. And each purple dot we see is a vertex or a spot where those triangles meet. So we get a measure of cortical thickness at every purple dot or every vertex. Our first analysis used measurements of cortical thickness, but we can also look at correlations of correlations. So in other words, how cortical thickness across brain regions are associated with one another. And this yields a measure of so-called structural covariance, which allows us to approximate brain-based networks using gray matter structure, in our case, cortical thickness. And then using these measures of structural covariance between, say, two regions, we can look at how this measure then correlates with the IQ adaptive functioning gap. There's a relatively large literature that has attempted to link brain structure and function to cognitive ability. And so we elected to take a hypothesis-driven a priori approach and utilize these 8 regions of interest in terms of analysis. This approach also reduces some of the concerns we might otherwise have concerning issues of multiple comparisons. In terms of our first metric of brain structure or cortical thickness, in each of the 8 individual regions of interest and their association with the IQ adaptive functioning gap, we found that for the typically developing youth, there was a positive association between the gap in cortical thickness and the right middle temporal gyrus, and a negative association between the gap in cortical thickness and a left parietal region. In terms of structural covariance between regions and the association of that covariance with the gap, we found that a smaller gap between IQ and adaptive functioning was associated with greater covariance in autistic individuals, which might be thought of as a metric of structural integrity between these left hemispheric parietal and frontal regions. In this talk, we showed that both autistic and typically developing youth show a gap between IQ and adaptive functioning, and the gap increases with age in this cross-sectional sample. Critically, however, in autistic youth, adaptive skills are far below what would be expected based on intellectual ability, or adaptive skills are more than 25 standard points below IQ. Research is therefore needed to understand the implications of this profound gap, for example, how the gap affects quality of life. The findings concerning gray matter structure in the gap are preliminary, but are suggestive of a larger gap between IQ and adaptive functioning being associated with structural covariance in hubs of the frontoparietal network that in studies of functional connectivity are associated with improved everyday performance in goal-directed decision-making, so better executive functioning. However, more research is needed in order to understand this association, and also more research is needed using additional neural markers to examine the brain and gap associations. Finally, I would like to acknowledge and thank our collaborators and our funding.
Hi, I'm Ben Yerys, and I'm going to be talking to you about executive functioning in childhood autism across settings and the cognitive spectrum. And when we think about measuring executive function in the real world, one of the most common measures that we use is informant report, in particular the brief or the behavior rating inventory of executive function, which has 8 underlying scales that then feed into 2 indices and a global executive composite. The 8 scales are inhibit, shift, emotional control, initiate, working memory, planning and organization of materials, and monitor. And when we look at prior research applying the brief into children on the autism spectrum, what we find is that it's one of the most reliable tools to sort of elicit executive function impairments. There's a great review by Greg Wallace from a few years ago, and there's also been a fairly well identified peak impairment on the shift scale, where there's sort of more impairment on that scale than any other across the brief. And the effect sizes in school-age children tend to hover right around a two of like with the Cohen's D. When we think about gaps or what's not known for sort of real-world executive function, we don't really have many studies or any studies looking at children that have a comorbid diagnosis of intellectual disability. Furthermore, almost all of our research that's been reported with the brief has been with caregiver ratings, which covers home and sort of in the community, but we know very little about school settings, and that's quite important since school may be one of the most demanding, if not the most demanding, setting for executive function support needs. So in the study, we had 2 primary questions. The first was whether the executive function profiles would be similar for those with and without an intellectual disability diagnosis. The second question that we wanted to answer was whether the challenges would be similar across the home and the school setting within each group. So in terms of our approach, we analyzed existing data that we already had on hand using the original version of the brief, and we had it both from schools ratings and home ratings. In terms of our participants, Here, this is sort of what they are in terms of sample size. We had 51 who were in the ID group and 182 in the non-ID group. They were matched fairly well on age and on gender ratio. Obviously, IQ would be different. It was sort of mandatory that everybody in the ASD plus ID group had a full scale IQ below 75. And they had to have some impairment in adaptive behavior functioning. And those with ASD without ID all had IQs greater than 75. You can also notice that the ADOS scores differed as well between the groups, with the ID group having slightly higher scores on the calibrated severity score. In terms of how we addressed our questions, we did 3 analyses. The first was that within each group, we would look at differences between the age and birth sex norms using a 1-sample t-test. The second question that we addressed was looking for a peak impairment. So could we replicate that shift impairment? And we did that using a one-way ANOVA. And then finally, to look at sort of similarity across settings, we used a Pearson's correlation to look at convergence, to look at sort of that inter-rater reliability. So this is the data for the I group of ASD plus ID. So what you can see here, first and foremost, is that with using a T-score scale where the mean is 50, all of these scales were significantly different across both settings from the sort of the published norms. The second thing that you can notice from this is that the inhibit scale with the school ratings, the shift scale for both settings, the emotional control scale for the school setting, the initiate scale for the school setting, the school and home setting for working memory, and the school setting for monitor, all of those are 65 or higher, indicating that those scales are in the clinical concern range. The third thing that I want to point out here is that if you look sort of across all the 8 scales within each setting, what you will notice is that they're roughly similar. There actually was no significant difference between the scales, so there was no peak impairment. And that, sort of is different than what we would see here in the group that has no ID. And that is, again, very similar in that there's like all the scales are significantly different from the sort of published norms. As you can see, there are a number of scales that have a score above 65, including both settings for the shift scale, the school setting for emotional control, the home setting for working memory, and the home setting for the monitor scale. And then finally, what should be somewhat obvious here is that, again, we've replicated our shift peak impairment, and this is actually true in both settings, that there's a score of 71 and 70, that both of those are significantly greater than all the other scales. So we then, as our third question, or our second question and sort of our third analysis, was doing these Pearson correlations to look at convergence or correlation across domains, or within domains across raters, I should say. And here what you can see is in the ASD without ID group, all of these bolded correlations, which is everything except for the initiate scale, all of those are significantly correlated with each other. When you look at the ASD plus ID group, you can see that there's not nearly as much convergence. Only the inhibit scale, the emotional control scale, and the organization material scales were significantly correlated. All the other scales were not significant. And this analysis, and actually all the analyses I've done, have been corrected with a Bonferroni's correction, corrected in terms of multiple comparisons. So in conclusion, this study is sort of unique because it's the first one to document that there's a real world executive function impairment across settings for an ASD plus ID group. It's also the first study to demonstrate that there's a peak shift impairment for those in the ASD without ID group in the school setting. So that had been demonstrated at the home setting, but this is the first to do it in the school setting. What this suggests is that children who have lower IQ scores may need broader executive function supports. And that may even differ by setting. So I won't go through all the different scales, but there were some scales that were impaired in the home setting and some that were impaired in the school setting, but not both. And so, it sort of says that, what we might take in terms of creating an executive function program for individuals with ID may look different than those without ID. And the other thing that I would say is, we saw really nice rater agreement for those with ASD without ID, that group had really nice agreement across almost all scales. In terms of future directions or further gaps to sort of pursue, I think, our definition of ID was somewhat liberal and inclusive. We said, full scale IQ had to be below 75, but that did not necessarily mean that verbal and nonverbal were both below 75. And so one would want to know if, our findings would hold if we had sort of really strict criteria for ID. Same thing was also true for adaptive behaviors, where we needed to see some impairment, but we didn't require all three sort of subdomains of the Vineland to be below 70 in order to be included in that group. In terms of another sort of future direction is that we didn't have great convergence across raters for the ASD plus ID group. So that sort of raises some questions about the psychometric stability of the brief. So that, could it be adapted or improved so that way it sort of better captures the experiences of individuals that are sort of on the, have IQs sort of below 75. And then finally, one of the things we were not able to address in this study for a number of reasons is we didn't really address any of the social determinants of health that may have influenced executive function ratings. And so that obviously would be really important in the future for understanding, what kinds of supports might be needed, as some might be direct intervention with the child, but other sort of interventions may be at the school level or in terms of, more systemic issues that may need to be addressed. I want to take a second to thank my funders, to thank the people that have helped support this research, including my center director, Bob Schultz. And with that, I will say thank you for listening.
Thank you for joining us today. We'll be talking about the neural and behavioral correlates of working memory in Down syndrome. Down syndrome is typically caused by a trisomy of chromosome 21. It is the most common genetic cause of intellectual disability, with a prevalence of approximately 1 in 1,200. Since the turn of the 21st century, increasing numbers of syndrome-specific pharmacological and behavioral interventions have been developed to target the cognitive underpinnings of intellectual disability. One area of active inquiry into the cognitive underpinnings is deficits in executive function. Executive functioning serves as an umbrella term for a set of distinct but related cognitive control processes that regulates one's thoughts and behaviors. One way of organizing executive functioning is Miyake's three-factor model, which has 3 core executive functions: inhibition, flexibility, and working memory. Research on inhibition in Down syndrome is mixed, with some finding skills to be consistent with mental age, while others have identified it as an area of weakness in the population. Research on flexibility in Down syndrome is also variable, with researchers identifying it as an area of both relative strength and relative weakness in the population. More consistently, working memory has been identified as an area of impairment. Specifically, deficits to both verbal and nonverbal elements of working memory have been identified relative to mental age-matched peers. Thus, working memory will be a target of the current study. Our study looks to answer 2 questions. First, what are the real-world correlates of working memory? Is it associated with everyday behaviors that are important for everyday functioning and quality of life? And second, what are the neural correlates that could serve as both biological treatment targets and biomarkers in clinical trials? With regards to working memory and real-world correlates, to date, there has been identified an association between adaptive function and caregiver reports of working memory. However, not much is known regarding the association between working memory and adaptive functioning when using performance-based working memory assessments. With regards to neural correlates, we know that in the general population, working memory is subserved by a network of regions, including prominent involvement of the prefrontal cortex. In addition, recent research also highlights the importance of the cerebellum in working memory. In Down syndrome, there's limited research on the neural correlates of working memory or really any other aspects of the Down syndrome cognitive phenotype. Prior research into the Down syndrome neuroanatomical phenotype has highlighted prominent frontal, particularly superior frontal, and cerebellar abnormalities. In addition, one prior study has linked cerebellar anatomy to short-term memory functioning in Down syndrome. Thus, The current investigation sought to examine relations between working memory performance in both adaptive behavior, as rated by caregivers, and brain morphometry, particularly the superior frontal and cerebellar morphometry. And we will be doing this in a sample of youth and young adults with Down syndrome.
Now we turn to our study's method. Our participants included 24 youth and young adults with Down syndrome. They were part of a larger study of brain development conducted at the National Institute of Mental Health in the Intramural Research Program. With regard to sample characteristics, the mean age of our sample was about 16, but with a large age range from 6 to 24 years. With regard to intellectual functioning, it was estimated using either the Differential Abilities Scale Second Edition or the Kaufman-Braith Intelligence Test Second Edition, depending on the age of the participant. The mean IQ for the sample was about 52. The range was from 24 to 77. A little less than half of our participants were female, and the sample was predominantly from a white, non-Hispanic background. With regard to procedures, the participants completed cognitive testing and magnetic resonance imaging at the National Institutes of Health Clinical Center in Bethesda, Maryland. Parents rated their children's adaptive behavior as well. With regard to cognitive testing, and in particular our working memory assessment, the Differential Abilities Scale Second Edition was used. In particular, the recall of digits forward and backward subtest was used, and the raw scores from these were totaled as our estimate of working memory. With regard to adaptive behavior, the ABAS-2, or the Adaptive Behavior Assessment System, Second Edition was used. Caregivers rated this on behalf of their children. With regard to MRI acquisition, the same 3 Tesla General Electric scanner with an 8-channel head coil was used. MP RAGE scans were acquired on all participants and were used for the current analyses. The details are provided here in the second bullet point. With regard to the processing of our structural imaging, we use the FreeSurfer Image Analysis Suite, and in particular, the Desikan-Kalani Atlas was applied to parcelate the brain. For the purposes of the current investigation, we will focus just on 2 regions of interest. This is just a clip from our introduction where we talked about the fact that the neural correlates of working memory and Down syndrome are poorly understood. If we turn to the DS neuroanatomical phenotype in a recent paper from our group using this same sample from 2020, we find prominent involvement of the frontal lobe, in particular the superior frontal gyrus, as well as the cerebellum in characterizing the neuroanatomical phenotype for Down syndrome. Thus, for the current investigation, the superior frontal gyrus gray matter volume and the cerebellar gray matter volume were the 2 regions of interest. With regard to our analytic plan for examining working memory and ABAS associations, stepwise multiple linear regression was used. In particular, the dependent variables included the ABAS composite raw score, so either the conceptual, social, or practical, and the independent variables were entered stepwise. In step 1, age was entered because we were using raw scores for these analyses, and then step 2 included our working memory assessment, the raw digits forward and backward from the DOS-2. With regard to the working memory morphometry associations, again, stepwise multiple linear regression was used. This time in step 1, age and sex were included. And this is because both of these are known to relate strongly to the developing brain over the age span studied here. In addition, in step 2, the raw digits forward and backward score was entered into the model to predict our 2 ROIs of interest. With regard to findings, first we turn to our behavioral findings. Now, I mentioned to you in the prior slides that regression was used in our analyses, and age or age and sex, depending on the analysis, were regressed out. Here, we're showing you residualized scores that show you the relationship between our working memory estimate, in this case, our digit span score, and our measure of adaptive behavior, here, the ABAS conceptual score. What we find is that digit span does add unique additional variance above and beyond age to the prediction of the ABAS conceptual score, adding about 23% variance to the model. With regard to the practical domain, again, we find a significant association after accounting for the effects of age, with digit span, the forward and backward, accounting for an additional 18% of variance in the ABAS practical score after age. Lastly, with regard to social, we do find a positive association. However, it fell short of significance, adding an additional 13% variance to the prediction of our ABAS social score. Turning to our neural correlates, first we look at the superior frontal gyrus volume. Again, we have our residualized scores here. And what we find is a significant positive association between superior frontal gyrus gray matter, and it was predicted by, again, age and sex, and then digit span forward and backward. In this case, the digit span score added an additional 9% variance to the prediction of the model after accounting for age and sex. Turning to the cerebellum, we also find a robust association. And what we find is that the digit span adds an additional 31% prediction to the variance in cerebellar gray matter after the effects of age and sex are accounted for. So now we'll turn to our discussion and turn back to Catherine Steffen.
In sum, we found a positive association between working memory and the ABAS practical and conceptual domains, but not the social domain. In addition, we found that higher working memory was associated with larger superior frontal and cerebellar gray matter volumes. These findings have important implications. First, working memory may be a target of intervention from both a behavioral and neural perspective. In addition, the superior frontal gyrus and cerebellum may serve as regions of interest in search for biomarkers of treatment responses. Finally, the association between working memory and adaptive functioning should be considered within the context of downstream adaptive correlates, including school readiness, academic achievement, successful transition to adulthood, job success, and other aspects of health. Our study was not without limitations. First, we utilized just one measure of working memory, and that involved both a short-term memory and working memory component, which may not be appropriate for all populations. Thus, future directions should look to utilize a developmentally appropriate battery of working memory tasks. In addition, we had a very small sample within the current study with limited racial and ethnic diversity. Thus, multi-site studies with samples representative of the US population would be helpful for future studies. And finally, our study utilized a priori neural regions of interest rather than having our data-driven whole brain investigations. Thus, whole brain investigations of both neural structure and function may be utilized in the future. We'd like to thank the individuals and families that made this research possible and our funder, the National Institute of Mental Health.
Hi, my name is Emily Kuschner, and I'll be presenting the final talk in this symposium. I am reflecting on the excellent presentations you've just heard on both brain-based and behavioral examinations of executive functioning in children with developmental disorders. And as we think about the great heterogeneity that we see in presentation of children with developmental disorders, the question has often become, who can do neuroimaging research? Can all children with developmental disabilities participate in these research studies, or can we only be looking at a subset of children? And I hope that over time, with some of the work I'm presenting and others across the field, the answer will become, with the right tailored supports, almost anyone can do neuroimaging research. I do want to recognize my collaborators and co-authors, funding sources, and most importantly, the families who have partnered with our work and really driven us to be better at what we do. So the work I'm going to present grew from the recognition that severely affected individuals on the autism spectrum are very frequently excluded from neuroscience and neuroimaging studies. The thought has been that cognitive and language impairments make it difficult to follow directions, sensory sensitivities make it challenging to tolerate the sounds and physical experiences of the neuroimaging scans, and that this is not sort of an appropriate fit. Our research uses magnetoencephalography, or MEG, to examine neural correlates, particularly of auditory processing in children who have autism spectrum disorder. And we've examined auditory processing differences in autism and associations with language impairment, but we've been left with the question of whether these brain behavior relationships were similar in children who never developed speech or have very little speech, often described as minimally verbal or nonverbal. But we were aware that this group of children on spectrum might fall into that category of sort of being perceived as unscannable. So we knew we needed to take a different approach, and we used a participatory research model to develop something called MEGPLAN, which is a clinical and technical protocol for obtaining MEG data. In this study, it was children who are minimally verbal or nonverbal and have autism spectrum disorder and are also often diagnosed with intellectual disability. So we used a participatory model with qualitative methods and developed this protocol with technical and clinical components. This MEGPLAN has been presented at Gatlinburg in the past and was just recently published in Journal of Neurodevelopmental Disorders. So you can go there for more information. But in particular, the clinical behavioral side can really be tailored and personalized and is tailored to each child and their family. There are 3 phases of MEGPLAN: an assessment phase, a plan and preparation phase, and then the MEG visit, with a stellar interdisciplinary team involved at every step of the way. Just as an example, MEGPLAN incorporates a lot of visual supports that we use both for preparation and desensitization and habituation leading up to the MEG visit, and then also to support management at the visit itself. So in our study with children who had autism and were nonverbal or minimally verbal, the data in the paper refers to that group, we had a 74% scan success rate. And that is scan success for what we describe as acquirable data, meaning the child is in the MEG machine, we have closed the door, and we can acquire data, because that's a first threshold to get to. Can we even get in the room and start recording? What we saw was that for children who couldn't reach that threshold, we saw lower, possibly sort of trends for lower nonverbal IQ, maybe lower adaptive behavior, but these weren't sort of slam dunk clear findings. And we saw that age, expressive receptive vocabulary, and ASD symptoms were not associated with scan success. And in our experience, we found that we were actually just not very good clinically at predicting who would be successful and who would not. And it didn't seem like the children who had the sort of lowest IQs heading into the visits were the ones who struggled the most. So we began to think about how we might take a more personalized approach and understand more about the individual differences in order to preview what supports were needed within the MEGPLAN protocol, see if we could determine the likelihood of scan success and be a little bit better at predicting, and maximize comfort and mitigate stress during study participation for children and their parents who are parents and caregivers who are very closely involved. And what we began to sort of observe and think more about was temperament and personality, and, passive versus more active kids who were just more reactive to things versus more laid back, and whether those sort of traits and qualities were things that helped guide how we approached the visits for the imaging and whether we saw success. So to give you a snapshot of the sample I'm going to talk about today, this is the broader study connected to the one I just referred to. So it's children who have autism and are minimally verbal or nonverbal, but also children, we had a nonverbal IQ matched neurodevelopmental disorders, genetic syndromes, a sort of heterogeneous group of children without autism. 8 to 12 years old, mean age was just under 10. Nonverbal IQ, vocabulary, adaptive behavior, all sort of mean in that intellectual disability range, but we did have pretty wide variability across the sample. So children received MEG with a CTF system, and it was a pure tone paradigm. So just listening to beeps, it's a very passive paradigm. They sit in the MEG machine. They can watch a video that doesn't have sound while they're in the machine, and their brain responds to the tones. And then what we decided to do was have parents complete the Carey temperament scale, which gathers information on the 9 categories of temperament. And version was based on nonverbal mental age, whether it was the infant version, the young child version, et cetera, to be most aligned with what children were doing in daily life. So what I want to highlight here and why I'm presenting in this symposium is that when we look at the different categories of this temperament scale, what we see things like amount of physical motion, but we see behavioral flexibility to the context, we see response to novelty, how well you manage extraneous stimuli, how much they can interfere with behavior, emotion regulation and intensity, mood, and reactivity to sensory information in the environment. And so many of these temperament categories have a very similar sort of flavor to the context of the content within the executive functioning domain. And as you've heard about with the brief, a lot of these categories and the questions kind of align with many of those constructs. So what I'm going to show you on the next slide are parent ratings on the Carey temperament scale, divided by group of children who had acquirable scan data with the MEG or never reached that acquirable point. So we never were able to turn on the machine for them. And these data are the category Z scores. So what you can see, the turquoise lines are the spots where the groups differed, acquirable versus non-acquirable. And those three categories were the distractibility category, how well children, how much extraneous stimuli interfered with behavior, the intensity category, the energy level of an emotional response, and then how parents rated them in sort of overall manageability. And the effect sizes were medium to large across these categories. So in this kind of preliminary look, the take-home points can be that we see increased difficulty for scan success in distractibility, intensity of emotional responses, and general manageability. And if we think about this, these are really EF domains. So as we think about the brain behavior relationships that my co-presenters have talked about in this symposium, there is risk of having more difficulty scanning children who may have more difficulty on those tasks. There's a sort of circularity that may limit use of technology in these methods. And so we really need to think about these temperament profiles and how we can personalize to have really tailored supports with an eye toward behavioral regulation, because that may be really crucial to ensuring that we are inclusive in our samples when we conduct these neuroimaging studies, thinking about executive functioning constructs across the wide range of developmental disabilities. I also just want to highlight at the end that there are some sort of cost and ethical concerns that we talk about a lot as a team. Imaging is very expensive. And so if you send a child for an MRI with no chance of them having successful data, that's a lot of money spent. But kids prove us wrong. We're trying to understand better who's going to be successful, but we want to reach a point where every child gets an opportunity because we think we can tailor and develop the right supports, provided they can stay safe in the environment, that we can support them to make that attempt at collecting neuroimaging and neuroscience data so that our results are generalizable and inclusive across the wide range of children with developmental disabilities. Thank you.
Thank you to all of our speakers, and thanks to all of you for being with us today. Before we take questions, we will end our symposium with a discussion by Dr. Lauren Kenworthy of Children's National Hospital.